Afternoon, everybody. Today is the second part of the story lecture given by Dr. Bruce Ames, a professor in biochemistry in Berkeley, a senior scientist in Children's Hospital, Oakland Research Center, and the discoverer of Ames test. And I have the honor and the pleasure of introducing him again. Yesterday, he gave a talk on the subject of delaying aging and optimizing health with supplement and diet. And today's topic of lecture is the metabolic tune-up to optimizing health. Dr. Ames, thanks again for coming, and please take up the podium.
So it's a great pleasure to be here. I've learned lots of things by talking to lots of the terrific scientists here. So my talk will overlap a little bit with what I talked about yesterday, but I'll try and give it a slightly different angle as much as possible. So 30 years ago, we published a paper. I used to teach a biochemistry lab for many years where I had the students isolate mutants of bacteria that would grow on a complex medium and not on a simple medium. And each student had to pick 3 mutants and do a little detective work and figure out what gene was damaged. And it was good to make the students think, but one kind of mutant kept on cropping up, and mutants I called KM mutants that had to do with binding sites. And so ordinarily, if you mutate the gene that converts threonine to isoleucine, you show up with an isoleucine requirement, and it grows on isoleucine. But some of those mutants will grow on pyridoxine or isoleucine. And you've mutated the threonine deaminase, which has a pyridoxal phosphate coenzyme, so that its binding for the pyridoxal phosphate isn't very good. And if you put in high pyridoxine, it can also make the enzyme work, and then you don't need the isoleucine. So it'll grow either on pyridoxine or isoleucine. And then some would grow on either threonine or isoleucine, and some would grow on either threonine or pyridoxine or isoleucine. Anyway, we published a paper on it with Esmond Snell and Beverly Gerard many years ago, but it stayed in the back of my mind for 30 years, thinking that this might be important in humans. And I think it's going to be. And I'll just start off talking about that. So you go into the health food store, and they sell B100 pills. Now, what's a B100 pills? It's 100 milligrams of all the B vitamins. Now, you need 1.3 milligrams of B6 every day. That's the recommended daily allowance. Why the hell is anybody selling a pill with 100 milligrams of B6? I thought it was crazy. Everybody in nutrition thought it was crazy. But this review we've just written says maybe it's not so crazy. So anyway, when we started working on aging, we found things that we thought were relevant to KM, to Michaelis constant and binding constant. And I set an undergraduate student to digging out the literature. And he was a terrific kid, or is a terrific kid, Ilan Elson Schwab. And between the two of us, and a little help from Eli Silver, who's another undergraduate, we cleaned out the literature and wrote this review, which Chuck Halstead kindly published in the American Journal of Clinical Nutrition. So it turns out you make a random mutation in a gene, you deform the protein, and if the protein has a coenzyme, 20% of the time or whatever, you've affected the binding constant for the coenzyme, which means you've lost activity, but it turns out you can reverse it because you can feed people huge levels of B vitamins and raise coenzyme levels 10 or 20 times. Now, why nature made it that way, I don't know, but it's true of all the B vitamins, which means that you can reverse the phenotype. And individual docs had stumbled on this, and this is the summary of the paper. And individual docs had stumbled on it, and often they'd say, yeah, it's probably a binding constant. Sometimes they'd even measure the KM. But nobody sort of looked around. And nobody dug out the literature from the nutrition community on what happens when you feed high niacin. Do you really raise coenzyme levels? Well, it turns out you do. It's all in the literature. And so we put it all together. And I think this review has a lot of implications, and I'd like to discuss that a little bit. So, for example, one of the rare genetic diseases is kids go blind with gyrate atrophy of the choroid and retina. And nobody knew why, but they knew it was a rare genetic disease, and some docs specialized in it. And then one day, they ran the blood of these kids on the amino acid analyzer, and there was a huge peak of ornithine. They said, aha, and then they fed ornithine to pregnant mice, and the pups came out blind. And they said, aha, and then they tracked it down to ornithine aminotransferase, which happens to be a mitochondrial enzyme, and that enzyme is defective in this genetic disease. And okay, so they learned what happened, but that doesn't do the kids any good, except some doc said it's a pyridoxophosphate enzyme, all these amine things that deal with amines of pyridoxophosphate. Why don't we, I'll try feeding them pyridoxine. So he gave a huge dose of pyridoxine to these kids, and they didn't go blind. So he published that, and from then on, anybody who got somebody with this disease gave them high pyridoxine. Some of them didn't go blind anymore. Some still did because it wasn't a binding constant. Depends on the mutation. And then someone else showed it was a KM. And similar cases like this came up in the literature. So we found genetic disorders with all these different compounds where there was reasonable evidence that you could reverse the phenotype by giving high levels of something, but mostly B vitamins. And then, now Pauling had this idea years ago. He said you should go to the mental institutions. All the neurotransmitters are amines. All the amines, metabolism of amines involves pyridoxophosphate. Why don't you just give high pyridoxine to everybody in the mental institutions? And maybe some of them have a defective Km binding constant, and we'll just empty out the mental institution. Well, it was a good idea. He was probably right to some extent, but he never gave any evidence that the thing worked. But I think it's not a bad idea. So Pauling had a terrific intuition. And then we found a number of polymorphisms. And now we're not talking about rare genetic disease. We're talking about a lot of people that where there's some phenotype and seems to be Km. So one is methylene tetrahydrofolate reductase, which changes the proportion of your methyl tetrahydrofolate to methylene tetrahydrofolate. If you're low in the methyl, it gives you heart disease. If you're low in the methylene, it gives you cancer. So we worked out why all that happens. I'll talk about that. But, I talked about last time that it saves your sperm, this mutation, and it happens in Northern Europeans. Well, it turns out it's at the FAD binding site, so high riboflavin, which raises FAD, ought to reverse it. And there are only two types of mutations, so in general, most of the people ought to be reversed. Sometimes you have many different mutations. But you can also reverse it with high folate B12 and B6, so maybe this won't accomplish any more, but there's some evidence in the literature that riboflavin does reverse it. Now, this is an interesting one, glucose 6-phosphate dehydrogenase deficiency. And that one, does anybody know what this is? It's fava beans. And it's, there was a wonderful artist in the Medici court, Giovanna Garzoni, who did these terrific drawings. There was a show at the National Gallery. And this is fava beans. And the fava beans was the main bean in the Mediterranean before the string bean came in from the New World. And Pythagoras, who is a Greek mathematician, started kind of a cult. And the first thing he told everybody is don't eat beans. Now, all the classical scholars thought he was a nut, and it went off his rocker. But he was actually very smart, because it turns out there are 400 million people around the Mediterranean that have glucose 6-phosphate dehydrogenase deficiency, And what that does is make you more resistant to malaria, which was wiping out all the kids. It was all malarial regions. And the reason it does is lowers that enzyme, regenerates glutathione. So basically, when you have a defective enzyme, it makes you a little more sensitive to oxidation, and the malarial parasite is hurt more than you are. So you're basically lowering your antioxidant defenses. But for every plant has 100 toxic chemicals it uses to kill off the insects. Those are the real pesticides you're eating. And the one in fava beans is vicine and divicine, which are powerful oxidants. So you can eat a meal of fava beans fine if you have normal antioxidant defenses, but if you have glucose-6-phosphate dehydrogenase, you get a hemolytic anemia, and you can die. So, and maybe 10% of the people on the island where Samos, where Pythagoras came from, ended up getting sick, and he figured it out it was fava beans, and that some people were sensitive, and so he made a rule, don't eat beans. So now it all makes a lot of sense. So he was smarter than the classical scholars. But so that, but if you have this mutation, not only do you have favism, smoking is a severe oxidative stress, aging is a severe oxidative stress. So these people have phenotypes, and there are other phenotypes that they have. And the people in India with this mutation, a lot of them, it's at the binding site for NAD. So it's the KM for NAD is affected. That's why you lose activity. So I'm just arranging with an Indian clinician who's working in my lab to get a clinical trial going in India to feed people high niacin and see if it helps. Anyway, we'll see whether it works. But I think there's a lot of mileage to be gotten out of all this. And one of the fruits of the Genome Project is to figure out who needs a little more B6 or niacin or whatever. Now, they use high niacin for heart disease, but nobody has a clue as to how it's working. Maybe it's raising NAD levels. So anyway, polymorphisms get to be, we're talking about a lot of people. Now, of course, an interesting one is aldehyde dehydrogenase, and that half of Asians can't drink alcohol. You all have Asian friends who can't drink alcohol. And it's all the same mutation, and you don't metabolize acetaldehyde to acetic acid very well, and it's at the binding site. So should giving Asians high niacin help? Well, the trouble is that KM is affected by 150-fold, and you can only raise NAD levels 10 or 20 times, so I doubt if it'll work, and a couple of Asian friends tried it, and they told me it didn't work. So anyway, we'll see on that. But maybe the other interesting things that'll come up. This is just the beginning. And so this is the one on that, and this is, okay. So now I'd like to get on to something else, but maybe it isn't so crazy the health food stores are selling these, and maybe some people feel better. And we don't have a clue as to what the RDA is for old people, because everything's done with young people, and maybe old people need some more vitamins, and maybe I need a little more B6 to turn me into a normal person. And now that might be a disaster, but anyway. So I think there's going to be a lot of individuality out. And what would be nice is to have a method to look at one person's cell and look at everything and say, your KM's a little worse for this or that. You need a little more niacin, or you need a little more of that to make you to have your biochemistry hum along a little better. But anyway, all that will come out in the future, and there'll be ways of doing that. But we're thinking about it, but we haven't figured it out. But I think for aging, it's going to have an impact, because I'm going to talk a little bit about aging, but we're working to see do high-dose B vitamins make human cells in culture live longer? And the answer is they do. And do they make Drosophila live longer? And the answer is they do. We're just getting the preliminary results on all of this, so I won't show you any data on all of that. They're modest increases, but all this biochemistry is getting fouled up in your mitochondria. Mitochondria do a lot of biochemistry. Membranes are getting stiffer, proteins are getting oxidized, aldehydes from lipid peroxidation are glomming on and shift bases to the lysine groups. So you get a higher level of oxidants and aldehydes and all these things. So maybe some of that lost activity is due to binding constant, and we know you can raise coenzyme levels in the mitochondria because in this review I point out there are 14 mitochondrial diseases that are reversible, so we know it's getting into the mitochondria. Anyway, that's all in the future. So if you look at cancer versus age in mice, in rats, in humans, it all goes up with something like the 4th power of age. So by the end of their 2 or 3 year lifespan, the rodents, a third of the rodents have cancer, a third of us have cancer. But it's all 60 million years of evolution. We've gone from short-lived creature like a mouse or a rat to people, and a huge increase in the age-specific cancer rate, and it's related to metabolic rate. Rats and mice have much higher metabolic rate. So cancer's under evolutionary control, and it's a degenerative disease of aging, along with heart disease and all the other things that come along. And why? Well, when the main thing damaging your protein and your DNA and everything is oxidation, because all that oxygen you breathe in is going to generate energy. You're combining, pulling electrons off of fat and carbohydrate when you're burning them for fuel in the mitochondria, and you add 4 electrons to oxygen, you're home safe to water. But superoxide, hydrogen peroxide, and hydroxyl rather, are just what you get from radiation. So living is like getting irradiated, and you're battering up everything. Now, and some years ago, we just had this epiphany. Your DNA is getting battered up all the time. What happens? Well, repair enzymes clip out the lesion, either nucleotide excision repair or base excision repair. Where does the lesion go? You pee it out. So people in Berkeley get high on all sorts of things, but was high on urine for a few years. And so we tried to measure in human urine and rat urine all the known radiation damage products, and you can find them all. Everyone, we looked for about 4 or 5 of them. Everyone we looked for was there. So you're constantly taking it out of the DNA and peeing it out. And we calculated there are 100,000 oxidative hits per a rat cell per day. So it's this huge battering up of your DNA. Now, you're making kilos of oxygen radicals per year because you're making a few kilos of ATP every day and all this electron transport. It's a pretty efficient machine, and we have DNA repair and we have all these other defenses, and they do pretty well because the steady state level in young rats and in old rats is pretty good, given that there are 100,000 hits per cell per day, and it's slowly creeping up, but that's a pretty good machine. But everything gets worse with age. Earl Statman has pioneered in looking at ketone carbonyl groups in protein. There shouldn't be any, but they slowly creep up with age in rats and in people. And if you take human cells, progeria or Werner's syndrome, progeria cells oxidize much faster, and Werner's cells oxidize much faster, and they're prematurely aging. So our DNA is being oxidized, our protein is being oxidized, our lipids are being oxidized. So you look in the different tissues of the rat, you can always find more aldehydes in the old rats than in the young rats. So, and then you see fluorescent material. If you do an autopsy on an old human and an old rat, you shine a UV light on the tissues, it fluoresces. That's called age pigment. So some junk is accumulating with age, and that's thought to be aldehydes cross-linking proteins and other things like that. So some years ago, Tori Hagen and Mark Shiganaga and I, it was, almost 10 years ago, we sat down and looked at what was the weak link, and we decided it was the mitochondria. Now, other people had come to the same conclusion. We thought we added a few insights. and it got us excited enough about it, we decided to work on it. So what's happening is you're doing all this metabolism in the mitochondria. An amazing amount of metabolism is going on in the mitochondria. And, of course, the Krebs cycle and all of that. And then basically you're pumping protons across the membrane to make a potential. It's a rechargeable battery. And that potential powers complex 5 here to make the kilos of ATP you make in your body every day. I've been meaning to sit down and calculate exactly how much ATP each of us makes, but do you do the molecular weight as the whole thing, or you're recycling the ADP part? Anyway, it's kilos to power your muscles and your brain and everything else. But you're bleeding off electrons, and then you're in trouble. But we have lots of ways of dealing with that, but mostly it works pretty well. But what happens to a mitochondria pouring out more oxidants? Well, there's a system for dealing with that. The lysosome eats up 10% of the mitochondria every day. And nobody's proven, but everybody thinks that it's when you have an aldehyde on the surface or some other indication of oxidation, those are the 10% of the mitochondria you take out every day. So even in neurons, you're turning over your mitochondria by the lysosome. But with age, Brunk and other people have shown the lysosome gets all clogged up. It's full of iron and cross-linked junk, and so it doesn't work very well, and then you get into more trouble. And so the system eventually just goes downhill, and you put out more and more oxidants. So this shows cardiolipin, which is a key lipid in mitochondrial membranes. It goes down with age. So old rats have mitochondria that don't work very well. They're spewing out more oxidants, and the cardiolipin is down, so the membranes are all different. And the membrane potential in old rats is very heterogeneous if you look at individual cells, and it's much less than in young rats. So all these things go wrong, and then the mitochondria just don't look right. These are mitochondria from young rats, and these are old rats. The structure just isn't the same. We're working with some electron microscopists to try and quantify all that, but it's turned out it's hard to do that, but he thinks he's finally solved the problem. And in the course of it, we got interested in carnitine, because some Italian company makes acetylcarnitine, and they support work all over Italy to work on acetylcarnitine, and some people in Bari, Italy, Gadoletta and Paradis, fed acetylcarnitine to old rats, and they said mitochondrial transcription got much better. So we had all these good methods, and we looked at that, and in fact, everything we measured in the mitochondria got better, except for one thing. They're still spewing out oxygen radicals. So the old rat mitochondria started looking much more like the young mitochondria in everything about oxidants, and then we found something else that works on that. So this shows carnitine. It's a beta-ene at one end, a carboxyl at the other end, a hydroxyl here, and you esterify the fatty acid, pump it into the mitochondria, burn the fatty acid, or put it into a mitochondrial membrane, and then put out a free carnitine and load it up again. So acetylcholine looks exactly like this, except for this acetic acid group coming off there. And then this shows cardiolipin, young rats, old rats with acetylcarnitine in the drinking water for a few weeks. You don't have to feed it very long, and already the rat mitochondria look much better. And this shows membrane potential, the old rats, and then the old rats fed acetylcarnitine, but doesn't do anything to young rats. And this shows, so then we went back to the drawing board to find something that would be an antioxidant for mitochondria. And Lester Packer's been saying for years that lipoic acid, which is a coenzyme for pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase, is a terrific antioxidant. So we tried that. We fed old rats lipoic acid. We actually fed this form. This is somewhat unstable, a dithiol compound. And it was terrific. It lowered the level of oxidants down to the level of the young rat. And so this shows this is oxidants being produced, young rat, and old rat fed lipoic acid. So then we tried the two together, and they really were frisky. So this shows vitamin C, rats can make it, but goes down with age, but the acetylcarnine lipoic bring it back up. And glutathione, the rats can make it, but it goes down with age, and again, this lipoic acid brings it back up, and the two together much better. And then aldehydes from lipid peroxidation, young rats, old rats, you get a lot more aldehydes floating around the cell and causing damage. And again, the lipoic acid brings it down, and the two together were terrific. So then we went into testing immune system. No, this was ambulatory activity. So you spy on the rats at night with a video camera hooked to a computer, and it tells you how much the rats walked around that night. They're nocturnal animals. Young ones are pretty frisky, old ones are pretty lethargic, but this peps them up. And this shows immune system. Immune system goes down with age, and this peps it, makes it better. So whatever we measured seemed better in these old rats. And then we knew we had to get into doing IQ tests in rats, but it's a big pain. There were years of headache trying to do all this stuff, but we eventually learned. So one is a Morris water maze. So you have a little pond with turbid water and a slightly submerged platform and visual cues all around. And rats can swim perfectly well, but they don't like to, so they make a beeline for the hidden platform. So the first day you put the rat in and you show them where the hidden platform is, and sits there for a little bit, and then you take them out. Next day you throw them in the pond, and the young rats remember and then make a beeline for the hidden platform. The old ones wander all around the place. So what that looks like is young ones and old ones. A huge difference between the old rats finding the platform and the young rats. And then acetylcarnitine lipoic make it better. So this is distance swum before they found the platform. Then people said, oh, it's just their eyes going out. You're making their eyes better, not their brain better. So the big visual cues, but it could be. But we tested that because there's another test. A psych professor at Berkeley, Seth Roberts, used to be at Harvard, and Skinner at Harvard developed the Skinner box. You put the rat in this box, and it presses a pedal, it gets a food pellet. So you can train it to do that. And what Roberts worked out is you make a sound or shine a light, and 40 seconds later, the rats have to press the pedal. You make them hungry for a day, and then you put them in, and you can train them perfectly well to press the pedal. But they have to remember this exact time interval. If it's 35 seconds, they don't get the food, and if it's 45 seconds, they don't get the food. They have to remember an exact interval. So it's a slightly different type of IQ test. But the young ones, is the green line, do it very well. Sound and light are identical. It's their brains. It's not their hearing. And then acetylcarnitine is the yellow, doesn't do much. The older the pink here. And acetylcarnitine doesn't do much, but lipoic does make it better in both. And the two together are better than either one alone. It's hard to see, but it's this purple there. I have to change those colors. So the brains really get better. And then we stained neurons in the hippocampus, and this is a monoclonal we developed some years ago that looks at 8-oxodeoxyguanosine in DNA, oxidized guanine. And so here are young rats, old rats, and old rats fed acetylcarnitine lipoic. So we did this with Carl Kottman at Irvine, who taught us how to do this staining. And we just assumed it was DNA, but then we looked at the whole cell, and the nucleus wasn't staining. It was all around, every place but the nucleus. So we thought, maybe it's RNA. We knew our antibody would react with 8-oxoguanosine in RNA. And in fact, it is RNA, because pure RNase takes away all the staining, and pure DNase doesn't. So you're staining RNA, we think, in the mitochondria, which is where you're getting the oxidants. And so what's the mechanism? And we think it has to do with binding constants. So the first odd thing is we did the enzyme kinetics for the enzyme that switches an acetyl group between CoA. So you have acetyl-CoA, and then you switch it on to carnitine, and then you pump it out. And so the binding constant for acetylcarnitine in this enzyme is different in old rats and young rats. So that was interesting. Here's the young guys, and here are the old guys. And then we feed it the acetylcarnitine lipoic. It makes the binding constant back to normal. Now, how can that be? Well, mitochondria are turning over, and the enzymes turning over. So you're getting new enzyme with a good binding constant, but it's changing it. And so the enzyme works better. And then the same thing is happening with the CoA binding constant. And and we can mimic this by taking pure enzyme you can buy from one of the chemical companies, and it's a pigeon breast transferase, but it's the same enzyme basically. And we add aldehydes, it inactivates it, and inactivates the binding constant, and you can prevent aldehyde inactivation by adding more acetylcarnitine to the, so the acetylcarnitine binds and then it prevents inactivation. And so we think that that explains it. If you just oxidize the protein, you inactivate it, but you don't affect the binding constant, you're affecting the Vmax. So at least we waved our arms and said we think we understand it. And in the literature, there are a lot of enzymes that lose activity with age, and some of them are Km changes, some are just Vmax changes. So, you're making all these aldehydes that can react with lysines and protein, and you're oxidizing protein. If it's a membrane protein, the membrane can get stiffer. So what about in humans? Oh, before I get to that, Tori Hagen has been working on the mechanism of lipoic acid, and lipoic acid, what he knew is that glutathione synthesis came up in response to the lipoic acid. Now, he did that when he was in my lab, and glutathione synthesis enzymes are under control of the phase 2 enzymes. Phase 2 enzymes, the pharmacologists like Paul Tallalay have shown there are over 200 enzymes that get turned on by a transcription factor at the antioxidant response element, and 50 enzymes that get turned off, and including glutathione synthesis, glutathione transferase, all sorts of things. And they've worked out the signaling system. There are 2 sulfhydryls next to basic groups, so they're very easily oxidized on a protein called KEAP1. When NRF2, the transcription factor, binds, everything's fine. But if you oxidize those sulfhydryls, then NRF2 pops off and turns on all these defense enzymes. So anything that hits a sulfhydryl, an electrophile mutagen that hits a sulfhydryl, a metal that binds the sulfhydryl, or if you oxidize it, turns everything on. And it turns out that the oxidized form, the disulfide of the lipoic acid, is acting as a mild oxidizing agent, and this then gets oxidized, this gets reduced, and you're turning on the system. So he just has a paper coming out in PNAS showing all this, and NRF2 goes down with age. Well, what about people? Senility in youth. So in a fit of enthusiasm, I called up my son in New York who works on computers, and I said, one of my students seemed to be changing old rats into young rats. And my son replied, oh, that's all very well and good, but you let me know when you do the next step, when you change old people to young rats. So your children don't let you get away with anything. And but there have been lots of clinical trials on the individual ingredients because they sell acetylcarnitine as a pick-me-up, sold over the counter in Italy. And they've done a lot of cognitive trials for cognitive impairment and mild Alzheimer, 21 clinical trials. And a meta-analysis by Montgomery that just came out says, yeah, it really works. And then there have been plenty of clinical trials with lipoic acid, which is used for the neuropathy of diabetes, and a meta-analysis, yes, it works. So there's experience with these things in humans, And so we took out a patent on the mixture, and the university got the patent, and I formed a company called Juvenon that licensed the patent. I put my stock in a foundation, so I have no financial interest in any of this. But the purpose of the company is to raise the money to do clinical trials on the mixture, and we have a big one going on heart disease, which will come out in the spring, I hope. And then we've got enough money now to do one on cognition. So anyway, I'm hopeful that this is just the beginning, but we're trying high-dose B vitamins, CoQ, a lot of other things to try and improve the mixture. And I think life expectancy and degenerative diseases, there'll be ways of dealing with them. So, and life expectancy, as you all know, gets longer every year. This is women, this is men, and every year it gets longer. Now, some of that's infant mortality, but if you eat right and you don't smoke, you have every reason to think that aging used to be a muddled field. There were very few people in it. You knew the dozen guys in the world doing good stuff in aging. But now lots of young people are getting into it, and there's room for lots. It's ripe, and we have all the tools. So you guys are going to live a lot longer than we will. Anyway, or you young guys. So I'm very optimistic. So the people involved with Zhangkang Liu, who is a postdoc and then a senior postdoc, now independent in my lab, who's been doing all the work in the lab on the acetylcarnitine lipoic after Torrey Hagen left. Torrey Hagen started it all and was in the lab a number of years, and now is at Oregon State. And when he left, we divided up the rat. He took the heart, and I took the brain. And he's done everything in the heart that we've done in the brain, and it really looks good. And then these are various other postdocs that have had a hand in it at one time or another, and we collaborate with them on the staining thing. And now I'd like to talk briefly about how you can age yourself faster. I'm sure you're all interested in that. But so in cancer, all the top epidemiologists think that bad diets are the big thing other than smoking. And obesity is some train wreck coming down the road. So we're not going to live longer if everybody gets fat because it's worse than smoking. There are going to be more deaths due to obesity than smoking. A third of the kids coming into Children's Hospital are obese. And the costs are going to be astronomical because years of diabetes and years of cancer and heart disease, any disease you want to name, asthma, it's all linked to obesity. And then I'm very interested in vitamins and minerals, and I'm going to tell you, if you don't get enough of your vitamins and minerals, you're aging your mitochondria faster. So, and people thought we don't have to worry about vitamins and minerals. They discovered vitamin C. We don't see any cases of scurvy anymore. They discovered beriberi. We don't see any cases of that anymore. Somebody gets carted off to the hospital if they really have an acute deficiency. But are we getting the optimum amount? And I think not, because there's a lot of ill health between the acute disease and the recommended daily allowance. Whatever vitamin or mineral you look at, and if you take less than half the RDA is a really low amount, and I'll show you it does damage in a number of cases. There are a lot of people out there, 10% or 20% of the population for each one, who's in trouble. And just women, menstruating women, are losing iron all the time, and a quarter of them aren't getting enough iron. A quarter of the women in the United States are not getting enough iron. Fernando Viteri tells me 20% of Berkeley women students aren't getting enough iron. And not getting enough iron ages you faster. Oh. And then we talk about zinc, 10% of the population is low, B6, 10% is low. Folate used to be very high, percent with low, half of the poor, but they're adding it to the flour now, so we've essentially eliminated that. And then B12 is quite high, and C is high. So this is biochemistry. All this is whirring around, and what people aren't thinking, they're worrying about a part per billion of pesticide residue, but they're not worrying that they're eating these lousy diets and they're not getting enough of the vitamins and minerals that are needed to run all this. You need 40 different compounds they're called micronutrients, the vitamins and minerals and essential fatty acids, to run all this biochemistry besides fuel. And we're just not, and the amount of oil you need in your car is not the amount to get it out of the garage, it's for the lifetime of the engine. And I think we've sort of forgotten about that. So I think a lot of ill health is due to not having our biochemistry tuned up. And now it's all cheap. None of this costs anything. You can get a multivitamin mineral pill at Costco for 2.5 cents. You can get a year's supply for about $12. And you should be taking it as insurance, but you should be eating a good diet too. So obesity, this is from The Economist. And so, you know, this is going to be really bad for the country. All you thin people are going to be paying all the medical costs for all the fat people for years to come. But is it just calories in and exercise out? I think it's more than that. This exercise is important, of course, but this is, yeah. So what else matters? Well, you take rats and you put them on a cafeteria diet, they get hugely fat. So some Brits did this experiment years ago. They took rats and let them eat all they want. And you put one set on Purina Chow or whatever they were feeding them, which is a good veggie-based diet, lots of vitamins and minerals and everything. And they ate all they want, but they didn't get fat. You take rats and you give them donuts and pizza and the cafeteria diet, and they ballooned up I think they were hungry all the time. So, and you test obese people, and there's several papers testing for lots of vitamins and minerals. They're at the bottom of the population for everything, because what they're eating is sugar. They're eating too many sugary soft drinks, too much donuts, too much potatoes. They're just not getting the vitamins and minerals. So I think one explanation is they're just hungry all the time. And there's at least some evidence in rats pointing that could be satiety could be more than just fuel. And for most of human evolution, we were grabbing a grasshopper here, a berry there, a piece of plant there. Calories were limiting growth. And in fact, the big brain developed to remember where the tree was, at what season that had that fruit that you could go get. Just getting enough calories was a big problem. But now, calories are dirt cheap, and you can get sugar with no vitamins and minerals. And when you look at what people are eating, and NHANES, you can get all this information out. The 10 leading sources of calories in the United States: regular soft drinks, cakes, sweet rolls, donuts, pastries, hamburgers, cheeseburgers, meatloaf, pizza, potato chips, corn chips, popcorn, rice, rolls, buns, English muffin, bagels, cheese or cheese spread, beer, French fries, and fried potatoes. A lot of sugary calories, but not much in the way of vitamins, minerals, or fiber. Now, fiber, this says, if you want fiber, madame, I suggest you eat the menu. Fiber is really important. Mark Shiginaga, my colleague, has a gut-centered view of the universe. So the gut, you have a column of bacteria in your gut, and fiber ensures you have good elimination of everything. And you need soluble fiber and insoluble fiber. You ferment fiber to butyric acid, which is the favored substrate of the gut cells. And what really hurts gut cells is a big dose of sugar or a big dose of fat when you don't have fiber. And then you start leaking lipopolysaccharide or bacteria through your system. At least this is Shigenaga's theory. And what it does is make you fat, because it makes you lethargic. When you have a flu, you get all this inflammation going on in your body. And obese people are very high in C-reactive protein, which is a measure of inflammation. And why are they high in C-reactive protein? Nobody knew. And he thinks it's just bacteria coming through and lipopolysaccharide coming through. And that interferes with beta oxidation, raises your blood lipids. It's making you lethargic and fat. And so not only are we short of micronutrients, we're short of fiber, and we're short of a number of other things like DHA, which is 30% of the fatty acids in our brain neurons. So we need, and these things are cheap. We need to get everybody up to snuff on all this stuff. And I think it's going to help the obesity problem. Now, we got interested in micronutrients partly because Jim McGregor, who's a cytogeneticist, had shown that folate deficiency breaks chromosomes in mice. And he came to my lab, and I put my graduate student, Ben Blunt, to working out why. And we came up with the idea that maybe if methylene tetrahydrofolate isn't working well, you don't convert uracil to thymine, and you start incorporating uracil in DNA. So Ben developed an assay for uracil in DNA, and you can put 1 million uracils in a human cell when you're short of folate, or in a mouse cell. So, and it turns out you break chromosome just like radiation breaks chromosome. Now, B6 is involved, and B12 is involved, and there are 2 pools of folate. So if this pool is, if you have both of these, you convert your homocysteine to methionine. If you're short of these, homocysteine accumulates, and that's a major risk factor for heart disease. There's a lot of interest in that now. And here, if you don't have this pool, you start putting uracil in your DNA, and every time you remove it by a repair enzyme, you make a nick, and so that breaks your chromosomes. So this has been associated with 4 or 5 different types of cancer: colon cancer, ALL, pancreatic cancer, and this has been associated with heart disease when this is low. And B6, folate, and B12 are involved in all this. But there's also a polymorphism which lowers this pool, it increases this pool at the expense of this pool. So it makes you more resistant to cancer and chromosome breaks, and more susceptible to heart disease. And it's in Northern Europeans. Now, it's easy to imagine Northern Europeans weren't getting enough folate. Folia, the Latin word for leaf, comes from folic acid is named after that because they isolated from 4 tons of spinach when they first isolated folic acid. And so you don't get your greens, you're not getting a lot of folic acid. So it's easy to imagine the Swedes weren't eating a lot of greens most of the year. And so maybe, but cancer and heart disease come way too late in life. And so one of my students just figured out why. We think it's the sperm. So you're also putting uracil in your sperm DNA and getting nicks and breaks. And that nature really cares about. So anyway, that's our hypothesis, why you're protecting your sperm at the price of getting heart disease. And radiation works by giving you clusters of electrons, and then when you repair them out by a uracil, by oxidative enzymes that do glycosylases that take an oxidative base off the sugar, and then you make a nick and stick this in, stick the right base in. But what hurts you is when you get too near each other, because then when you're repairing out about the same time, the chromosome falls apart. And that the radiation biologists think that's the dangerous part of radiation. Well, by the time you put 1 million uracils in your newly synthesized strand, and you have 70,000 or so oxidative damage from just living, then you get the same thing. And we have a paper that just came out in FASB Journal where we compared folate deficiency with radiation. Radiation isn't even in the same league with micronutrient deficiencies, and people were talking about spending $6 trillion to clean up Hanford, where little bits of radioactivity got into the Columbia and other sources, but they're awfully tiny amounts. You could give everybody around Hanford a vitamin pill, you'd be way ahead of the game, and $6 trillion saved in money. So, but epidemiology is very hard, but intervention studies are relatively easy. So Terry Schultz up in the state of Washington works on B6, and he takes 12 people, puts them on a defined diet, and puts them on half the RDA of B6 for a month, then the RDA for a month, and then looks at what happens. And so we're collaborating, we're looking at the DNA damage, and he's doing the intervention studies. Here are the group, I think it was about 10 people. Here are the people at half the RDA of B6. Here's the RDA for B6. We're measuring the COMET assay, which is using the COMET assay, which measures nicks and breaks in your DNA. Here's 2 times the RDA, and here's about 7 times the RDA. Not much here, but you don't need a statistical analysis to see that at half the RDA for B6, you're breaking your chromosomes. And so we're just doing the uracil, and then we'll write all that up. And this shows that you can get uracil in human sperm DNA, and when you take mice and make them deficient in folate, the sperm count drops. And we looked in humans with the USDA, Bob Jacob at the USDA, and folate deficiency correlates with both sperm count and sperm quality. So your sperm count goes down, your sperm quality goes down when the methylene pool goes down, but it's not correlated with a methyl pool, which would fit with a uracil incorporation mechanism. And this shows B6 deficiency, less than half the RDA is in red. A lot of people are like that, and the poor are always much worse off. So you look at blacks, Hispanics, poor, uneducated, they're all kind of tend to be mixed, hard to disentangle. They're at the bottom of the list. And obese. Obese people, no matter what their poverty or whatever, are also at the bottom of the list. And so we got interested in iron because Fernando Viteri, who's a professor in nutrition, has been interested in iron and why you have all these iron deficient people and how much iron you should give them. And so his student, Mitch Knudsen, and my postdoc, Patrick Walter, looked at the whole range of iron in rats. And so too much iron is bad for you, and it's bad for the mitochondria. You get mitochondrial oxidative damage, you get oxidants produced, you get inefficiency in the mitochondria. But too little iron is a disaster, and that's the 2 billion women. And Hani Atamna, a wonderful postdoc in my lab, worked out why. So heme synthesis is mainly done in the mitochondria. You're using iron sulfur clusters and heme are the 2 main sources of iron, and both of them are very important in mitochondria. So succinyl-CoA comes off the Krebs cycle, and you need 8 succinyl-CoAs to make porphyrin. So succinyl-CoA and glycine with a B6 enzyme makes ALA. And then ALA is pumped out, and there's an enzyme that makes a dimer of ALA. And 4 of those dimers then get to make a porphyrin. You pump it back into the mitochondria, and you do a few more steps, and then ferrochelatase inserts the iron. And what he found is when you interfere with heme synthesis, the first thing that goes out is heme A, and complex 4 goes out. So complex 4 has a heme A in it, which is a highly decorated form of heme, and it's the first thing goes out when you interfere with heme synthesis. And what that does is just fill the cell up with oxidants because complex 4 interrupts the chain of the electrons. And so Atamna has been doing a lot of nice stuff on that. And so here's complex 4, and now you just start pouring oxidants out. So you can measure this in lots of ways. And basically, you're knocking out your mitochondria. And which organ is most important? Well, the brain is very metabolically active. It's a high percentage of the oxygen you breathe in is used directly by the brain, 20% or so, even though it's not 20% of the weight of the body. And the mitochondria in the brain tend to go out when you interfere with mitochondria. Alzheimer's has been linked to mitochondria. Parkinson's has been linked to mitochondria. Neurodegenerative disease have been linked to mitochondria. So we think what you're doing when you don't get enough iron or you don't get enough B6, you don't get enough copper, you don't get enough pantothenate, you don't get enough biotin, one after another, we're linking these things to heme synthesis. You knock out complex 4, and you start filling the cell up with oxidants. And Atamna and I had a paper in PNAS where he showed that all the things that happen in Alzheimer's or neurodegeneration are the same things that happen to cells when you make them heme deficient. Your loss of complex 4, accumulation of iron. This is if you do it by ways other than iron deficiency. oxidative stress, APP aggregation, Alzheimer precursor protein, NOS increased, et cetera, et cetera. So how many of these things can be contributing to Alzheimer's and neurodegeneration? I wouldn't be surprised if a lot of deficiencies are, because they're very common in the population. I think half the population is probably too low in something. You look at, and this shows iron deficiency, menstruating women mostly. That's why they sell 2 pills, one with iron for the women and one without iron for the elderly women and the men. And again, black women are much worse off. And this shows biotin deficiency fills the cell up with oxidants, and that we know is involved in this heme pathway by regenerating the succinyl-CoA. And Emily Ho, a Canadian postdoc lab, works on zinc, and she's been showing that zinc deficiency fills the cell up with oxidants. We haven't proven yet where it's coming from, but I suspect it's also knocking out the mitochondria. So zinc deficiency gives you a lot more oxidants, and zinc deficiency really batters up your DNA, and then you can't deal with it because it's knocking out P53 and it's knocking out APE and it's knocking out antioxidant defenses. You get a quadruple whammy when you're low in zinc. And so why is zinc deficiency filling up the cell with oxidants? Well, next week we may know, we're doing the experiment now, but an enzyme that converts 1 ALA to the dimer of 2 ALAs has 8 zincs in it. And we suspect that's what may be going out, but we'll know that soon. And this shows zinc deficiency. Again, a lot of people so basically most adults, you're talking about 10% of people are low in zinc. And again, the poor are worse off. So Mark Twain said the main distinguishing characteristic between man and the lower animals is the desire to take pills. So he was a little suspicious of popping all these pills, and the whole nutrition community hates the idea of vitamin pills. They say, why tell people to take vitamin pills? They'll just not eat any fruits and vegetables. But they've been trying to get the poor to eat fruits and vegetables for 20 years with very little success. And I think what you should tell them is all the things that go wrong with them, including their brains going out, if they don't get their vitamins and minerals, and take a multivitamin mineral pill as insurance and eat a good diet besides, because we don't understand everything. And you need the fiber, which you get from your fruits and vegetables, and you need the DH, the linolenic acid, and you need to eat some fish, et cetera, et cetera. So, and all this is cheap. It's just will. And I think there's going to be a big public health advance if we can convince people that that's the right thing to do. And, you know, it doesn't cost it. We're spending all our money on chasing after pesticide residues or things that are highly implausible and not going to be serious things in any case, and I think it doesn't make any toxicological sense anyway. $150 billion a year is the EPA budget. We should be putting that into nutrition and getting people healthier. And so there's a lot of work to do. And these were some of the people involved. Thank you. Happy to answer any questions. I can't see with the light.
Do you think that homocysteine may be toxic, or is it just an indication of folate, B12, B6 deficiency?
No, people think homocysteine is toxic. They don't quite know why, but it seems to damage endothelial cells. And homocysteine looks like an amino acid, gets into tRNA synthetases, and makes a cyclic form called homocysteine thiolactone, which will react with protein. So that might be the toxic molecule. Nobody's quite sure. But it's certainly, they think it's the homocysteine itself. Yeah.
You mentioned neurodegenerative disorders. I wonder if anyone has investigated the effects of
I don't know anybody who's looked at that. Yeah. Is there someone? Yeah.
Is there a major effort done by NIH on caloric restriction if you reduce your calories by 30%?
Right, yeah.
That's the main factor for longevity. Do you want to comment on that?
Yeah, caloric restriction works in mice, it works in rats, but people, they don't tell you that mice and rats don't breed. They cut out reproduction when you starve them for calories, and they're awfully angry. They'll bite you if you get even near them. So I don't think it's going to work in people. But I think it will learn a lot about mechanism, but it's not a practical intervention. Maybe in Roy Walford, but not in the rest of us.
Could you say, if you mentioned passing something about biotin being sensitive away?
I'm sorry, biotin?
Yeah, your mechanism for...
Oh, yeah, biotin. So the role of biotin's are basically a mitochondrial coenzyme, just the way lipoic acid is. And what biotin does, it replenishes ingredients that come out of the Krebs cycle to use for metabolism. So you're taking things out for amino acids and all that, but you can recycle those and put them back in. The one thing that comes out and doesn't come back in is the porphyrin for heme, and you're making a huge amount of heme. So that's the main drain out of the Krebs cycle, and biotin's role is to replenish that. It's called anaplerotic for the Krebs cycle.
There's a lot of advertising on HGH supplements.
Yeah.
Do you have any comments about that? I don't know if they're useful or not.
I don't know about that, but there are a lot of things. The health food store has a wonderful array of substances that you can buy that'll keep you younger, make your sex life better and whatnot. So the only thing, I'll tell an anecdote, DHEA is a hormone. It's the main steroid hormone in the blood, and it's a precursor of testosterone. And it goes down with age. So lots of old men go into the health food shop and fill up on DHEA. And I have a friend who's a professor at UC San Francisco, and she was doing a clinical trial on DHEA, and I met her at a cocktail party. And she said, Bruce, guess what? 38% of the men who took DHEA pills said their sex life is much better. And I said, wow. And she said, Bruce, guess what? 39% of the men who took placebo said their sex life was much better. So you have to be careful about all this because there's a lot of anecdotal.
I was just wondering if you take your own supplement.
Yeah, you never guess I was 100 years old, but I do take Juvenon pills. Some people, we get a lot of letters from people who say they feel terrific, but I didn't feel a thing. And my rough idea is maybe 2/3 of people don't feel anything and 1/3 of people feel terrific. And the people who feel terrific are the ones who are a little chubby and pre-diabetic and all of that. So it seems to, we've gotten a lot of letters saying it lowers your blood pressure, but that's all anecdotal. Anyway, the clinical trial will test all those things. Oh, doses of vitamins? Oh, that, yeah, look up the Juvenon website and they'll tell you all that. Yeah. So, vitamins, I don't advise anybody take huge doses of vitamins. I think you tell people selenium's good and 5% of the population are going to go out and poison themselves with too much selenium. The metals, it's really tricky. Too much zinc is a risk factor for prostate cancer, but too little zinc is a risk factor for prostate cancer. You just don't want to overdo it. So take a multivitamin mineral. If you want to experiment with a B25, B50, B100 pill, work up and be careful. That's fine, but it's not good to tell the public to go pop all the vitamins they want to take. Russell? Magnesium. So we're taking every vitamin and mineral, making human cells in culture limiting, and then watching what happens. So David Killalay in the lab is just doing magnesium, and it looks very interesting, but we haven't repeated it yet, so I can't, I don't want to talk about it till he really pins everything down, but it looks interesting. And magnesium, there's a lot of deficiency out there because people aren't getting it. You get that from your fruits and vegetables. If you eat your 9 portions of fruits and vegetables a day, you're getting enough magnesium, but most people aren't. Yeah. Okay. Good. Thank you.